Okie dokie. I believe we're currently recording. All right. Good stuff. So, like I said, 95% of recommended textbooks you do not need to have. The most common ones that people actually have, especially in physical form, is anatomy textbooks or atlases. So those are the ones that you want to be having because those are like just the ones that are convenient to have, and they're the ones you're going to have the most use out of. So yeah, do all the boring admin modules, your like recommended stuff, all of the stuff they get you to upload to like PebblePad and whatnot. Obviously it's boring, but you do need to get them done because they'll lock course progression behind it, obviously. And yeah, ask questions. So both in this presentation and in your lectures and your worksheets and everything, they're all here to help, and they're all happy to answer questions about stuff. The other thing we can say, absolute 100% guaranteed success, come to PSPlatypus. We're definitely not biased at all. So we'll have a session every week. This week we're at 11 a.m. because you guys had that the whole presentation thing. After this week, we're going to try and move to 10 a.m. on Mondays, depending on if that fits with your timetable. And yeah, have fun. This is like year 1 and 2 is quite chill for med school, as relative as that might be. So yeah, enjoy yourself, take your time and make sure you have fun with the content. So you guys have come from an intro webinar, so I believe you know a little bit about this, but we've basically got 4 themes in medicine. So theme 1 is ethics, medical law, medicine of the mind, and HEP. So that's psychiatry, how not to get sued as a doctor, and like HEP is a bit like meditation, spirit and soul, sort of like well-being stuff, basically. Theme 2 is a single subject. It's called health, knowledge, and society. And that's all of the big picture stuff. So like indigenous health, refugee health, how the health system works, how the pharmaceutical industry works, all of that big picture stuff. I don't know if you guys have met Maitri yet, but if you got him, he's a great lecturer. You'll never find a man more passionate about his subject. Theme 3 is where we have all of the basically the stuff that you thought you would learn in med school when you're like year 9 or year 10. So this is your anatomy, your biochemistry, your pharmacology, immunology, all of that stuff, which is like very book knowledge-y. So that's probably, not definitely, in fact, not probably, that's definitely the biggest theme. That's got a lot of content. The bulk of your lectures, your workshops, everything will be on theme 3. And it also makes up the bulk of the exams. And theme 4 is clinical skills. So that's the one where we talk about like interviewing people, like asking people, taking diagnosis and that sort of thing. Although you won't get to do the really interesting stuff until a few years later. But yeah, we're slowly building up the skills on being an actual doctor. And our assessments are all like sort of a mix of those. So the exams cover the full course. So they're themes 1 to 4. And you have 2 of those at the end of each semester. And these are like pretty standard. We have a like really long multiple choice section, and then you have a short answer section at the end. Excuse me, because of COVID, they shifted ours online last year and they took out the short answer section. I don't know if they're going to try and do those in person for you guys this year. Richard might have told you something about it. But either way, it's pretty straightforward. This is the sort of stuff you're used to. You also have a bunch of written assignments. So those are generally only on themes 1 and 2. You might very occasionally have something on theme 3, but it mostly sticks to those ones. There are maybe 3 or 4 of those per semester, and some of them are group things and some of them are more individual. So like you do them by your own. And these are quite varied. But the good thing about those is that though you do get a mark for them, in the end, they ultimately pass fail. So they don't really count towards your WAM, only the exams and the CIPs.
The axes are pretty.
Thanks. The CIPs, they're like sort of it's like a big grid where you get a bunch of different topic areas on one side and a bunch of different medical conditions on the other side. You have to match them together. You'll hear a lot more about them from Richard. All you need to know is you only get 1 per semester and they're pretty straightforward. And the last one is probably the most like medi of them, which is a simulated clinical encounter. So like you basically pretend that you're an actual doctor doing for you guys like a medical interview or something like that and doing an examination of a patient. So in a non-COVID world, you'd have 3 or 4 per semester. We don't know how they're going to try and do them this year, but keep an eye out for those. Okie dokie. Do we have any like immediate questions based on that stuff before we jump into our first major topic today, which is cells? All right. If something comes up later, as I said before, feel free to chat in the chat and we'll deal with it at some point. Okie dokie. Very first topic we have today is cells. So this is covering a lot of the stuff that you would have done in either VC 3-4 Bio or the Bio Bridging course, but we also go a little bit beyond that and we could talk a little bit more in detail. So the very foundation of what we talk about cells is cell theory. And you definitely have learned about traditional cell theory, which is your three basic maxims. So all life is composed of one or more cells. The cell is the basic unit of life. So if we have a mitochondria or other organelles or that sort of thing, then we can't consider them living. And all cells come from pre-existing cells. Modern cell theory is basically, we looked at all of those and we came up with a few extra dot points because it wasn't comprehensive enough. So the first thing we found out was cells contain genetic information, which are passed on during division. That's how we get all of our genes and inheritance information. We find that cells are structurally and chemically similar within a species. So even though you have really varied cells within your body, like a smooth muscle cell to a neuron or something like that, they're still basically similar. They've got the same DNA, they've got the same moving parts, that sort of thing. And the final part is that energy flow occurs within cells. So that's basically saying cells are able to take things like glucose, food, and turn them into energy for the cell to use. So modern cell theory is not a replacement for traditional cell theory, it complements it. So it's basically saying this and. We have two major types of living cells, prokaryotic cells and eukaryotic cells. Your prokaryotes are your very simple organisms, basically, bacteria and archaea, and your eukaryotes can be quite a wide range. So some of the big differences between them are obviously that the prokaryotes have no nucleus, they just have a singular circular chromosome and some plasmids hanging around in the cytoplasm, whereas eukaryotes have a defined nucleus, and that becomes very important for the functioning of the cell. Prokaryotes are generally smaller than eukaryotes as well. If you see a micrograph image or something of those, then you'll find out they really don't compare in size. Organelles are a major feature of eukaryotes. Prokaryotes do technically have organelles, but they don't have any membrane-bound organelles. So eukaryotes are able to section off little parts of their cytoplasm and turn them into their own little environments. So basically everything that isn't bacteria and archaea is a eukaryote. And we know that prokaryotic cells can only be unicellular. Eukaryotic cells can be unicellular, but they're not always unicellular. We also have multicellular eukaryotes. So we've got a bunch of different terms that we talk about when we actually talk about a cell. So the cell membrane is our phospholipid bilayer. You might have heard of that before. We'll go over it a bit more in a sec. But we've got that membrane which controls entry of things to the cell. And that's going to be a major factor in how we control that and basically how the cell intakes things and how the cell gets rid of its waste. Organelles, they're specialized subunits of the cell. And so each of them does a specific function. And each of those functions is going to be, it needs a certain set of conditions to function most effectively, which is why we bother with them in the first place. The cytosol is the liquid within a cell. So that's just the fluid, it's quite watery. Inclusions are the things which aren't organelles and aren't cytosol, but they're floating around. So you might find things like protein stores, starch stores, all of that. The cytoplasm is what we get when we basically take everything in the cell except for the nucleus. So we have the cytosol, we have the inclusions, and we have all the organelles. Now, the plasma membrane that we have coating the entire cell is what we call a phospholipid bilayer. A phospholipid is a type of lipid which has a phosphate head and two fatty acid tails. You might have heard the term amphipathic to describe them because they have a head which is hydrophilic and a tail which is hydrophobic. So when we put them between two hydrophilic environments, i.e. the extracellular fluid around the cell and the cytosol within the cell, they orient into two layers with the tails pointing inwards. So they have a hydrophobic environment in the inner layer, in the core, which prevents large hydrophilic molecules from getting in. Now the small ones can fit through the gaps with things like CO2 and H2O and some amino acids, just little molecules which are hydrophilic, and if they hit any of those phospholipids, they're going to bounce back, but you can usually get through without hitting them, basically. So we often describe the plasma membrane as a fluid mosaic, and we call it fluid because it's sort of like a film on the surface of a water. All of those phospholipids, those proteins, all of the things we find in them, they can move and slide past one another. And we call it a mosaic because there are many different components in the plasma membrane. So phospholipids are a main component. They make up the bulk of the plasma membrane. We have cholesterol, which sits inside there and regulates membrane fluidity. So if the membrane becomes too spread apart and too active, cholesterol sort of pulls it back together. If the membrane starts to get too packed together and inactive, cholesterol pushes it apart, so it regulates it. So proteins, they can either go all the way through the membrane, and we call those integral proteins, or they can sit on one side, and we call them peripheral proteins. If I go to the previous slide, you can see near the bottom left there, we have a peripheral protein that only sits through one layer, and we have an integral protein next to it, which goes all the way through. So these actually have quite a few different functions. So they can serve as receptors for signaling molecules that we get from other places in the cell, and they can be transport methods for different substances through the membrane. And this can be either channel or carrier proteins. We also get carbohydrates. They don't exist in the cell membrane on their own, but they are attached to either phospholipids or proteins. And they're going to either identify the cell to other cells, or they're going to serve as receptors. So their main role is communication. All right, the nucleus is arguably the most important organelle, and we use it to contain the cell's genetic material in the form of DNA. So it's coated in a double membrane, and we'll talk a little bit more about how that double membrane interacts with other organelles, such as the endoplasmic reticulum. The actual name we give to DNA inside the nucleus is called chromatin, because DNA doesn't exist on its own. It's not just a sort of spaghetti mess floating around. Instead, if we look at that bottom right, we can see that DNA finds itself wrapped around histone proteins. We actually have what we call a nucleosome, which is this little package of histone proteins wrapped around with DNA, and then those coil into the really, really big chromosomes that you see. And when we need to use DNA, we actually need to unpack it from that, because we can't use the DNA while it's all wrapped up and unable to separate. So we use the term heterochromatin for the DNA while it is tightly packed and unable to be used. But then when we need to actually turn it on, we turn it into euchromatin, which is much more loosely packed, and we unravel a bit of that structure so we can get access to the genes we need. We've also got the nucleolus, which shows up as sort of a dark patch. And that's the site of ribosome synthesis. So that's where we make all our ribosomes, which are our little protein factories we use in the cell. We've also got something called the lamina, which is a fiber network within the nucleus, which sort of gives it some structure and serves some other purposes. You don't need to know too much about this. And there's another thing called the nuclear matrix, which is a lot more theoretical, but we think it's pretty similar to the lamina. Right, this is a micrograph image of the nucleus on the right here, which just basically means a photograph taken through a microscope. I'm going to try and give you guys a few of these, because one of the things that you'll have to do throughout med is identify different organelles and cell features under a microscope. So one thing that they might not have told you in VCU Bio is that we can actually have a lot of different numbers of nuclei in a cell. So although most cells have one nucleus, we can also have cells with no nucleus. So red blood cells and keratocytes, i.e. the ones in your skin, those don't need a nucleus to do what they do. So they don't have one. On the other hand, we have skeletal muscle, which needs multiple nuclei. That's sort of like a huge, big fiber. And so it needs more than one nucleus in order to effectively communicate with the rest of the cell. We can also have fragmented nuclei. So these are ones which sort of exist in multiple joined pieces, and those exist for neutrophils. Cells can also end up with either multiple nuclei or no nuclei because of errors in cell division, and we'll get into that a little bit later. All right, mitochondria, probably the organelle that you are most likely to know, and this is the site of aerobic respiration, which is where we produce the majority of energy that the cell is actually going to use. This is a double membraned organelle. The outer membrane is very smooth and forms the actual surface of the mitochondria. The inner membrane is very folded and forms these little sort of infoldings called cristae. That's very important for cellular respiration, which you probably learned about and which you'll learn about in a little bit more detail later in the semester. Mitochondria are inherited maternally because basically you are made from an egg and a sperm. The sperm only donates its genetic material, whereas the egg keeps all its organelles. So you only get the mitochondria from your mother. And that's important because mitochondria have their own DNA and their own ribosomes. So we can actually trace people's genetic material through their mother's line by looking at the mitochondrial DNA. And of course, the reason why they have their own DNA and their own ribosomes is because they were originally bacteria, and they were eventually integrated with eukaryotic cells by endosymbiosis. So a eukaryotic cell absorbed one of them and decided, hey, this isn't so bad. You can make energy for me and I'll provide a home for you. And they've been together ever since. This is a little micrograph image of a mitochondria. They can be little circles like this, or they can be really fat and long. There's a lot of them in between, but basically if you see something which looks like a circle with a bunch of lines across it, it's probably a mitochondria. All right, we've got ribosomes. These are technically not a proper organelle due to their lack of a membrane. They just sort of free float in various places. This is where we have protein synthesis. So mRNA comes from the nucleus and hooks up here and makes proteins. We have, it is itself made of RNA, which we call ribosomal RNA or rRNA, in a large and small subunit. So the large ones here, small ones there. And they sort of clamp together around an mRNA strand. And we can find these in a number of different places, so either floating freely in the cytoplasm or bound to the RER, the rough endoplasmic reticulum. And in both places, their one role is to make protein. So we've got a micrograph here. You may not see them in a regular micrograph of the cell because they are really small compared to the other organelles. Right, we've also got the rough endoplasmic reticulum. So this is where we make proteins, usually for export from the cell or for use in the plasma membrane. So it's got a bunch of ribosomes studded on it, and they're the ones that make the proteins, and then they put the proteins inside the RER where they're modified and packaged. So the actual structure of this is like a bunch of flat sacs, and its membrane is actually continuous with the outer nuclear membrane. So if you sort of imagine it, the inner nuclear membrane is almost like a sphere wrapping around your genetic information. The outer membrane is usually a sphere, but then at certain points it bulges out and makes these big sacs, and that's how we get the RER. We also have ribosomes embedded in the surface of the cell, which actually allow it, sorry, I said that already. That's how we get our protein production, and they chuck them into the actual lumen or the inside of the RER. And the reason that's important that we modify and we package those proteins is because we can turn them into vesicles. A vesicle is basically just a little sphere surrounded with a plasma membrane, and that's the way that the cell transports things around. So proteins, lipids, various other products. And that's super interchangeable. So our little RER here has a plasma membrane which can butt off into a vesicle, and it can send that to any other organelle which also has a plasma membrane and can absorb those vesicles. So it's a really interchangeable method of transporting things. We also have this smooth endoplasmic reticulum. So it's a bit like the RER. It's in fact joined to the RER, but it has no ribosomes because it doesn't make proteins. So this one looks a lot more tubular. So it's been described as the difference between spaghetti and lasagna. I think I was a bit hungry when I wrote these slides. But you can see here we've got very flat sacs for the RER and very small tubular, circular almost shapes. And you can see that in the micrograph down here as well. So they can actually serve a lot of different functions depending on the cell which they're present in. So in certain cells which need to make lipids, they're going to make lipids, fatty acids, steroid hormones, all of that sort of thing. And in places, especially the liver, they serve to detox things. So things which are potentially harmful to the body, they're going to be deactivated here in the smooth endoplasmic reticulum. We also have a function as the storage and release of calcium ions. So this happens only in muscle cells, and we call the SER a sarcoplasmic reticulum in those cells because it has that very specific purpose. And you'll learn more about that when you do muscle anatomy. Right, we've also got the Golgi apparatus, which is the sort of the post office of the cell. And so its role is to receive a cellular product from various places. It could be in the SER or the RER. And then it modifies them itself, and it packages them, and it eventually exports them from the cell. So it's also made of flattened cisternae, like the RER, and it has two faces, a cis face, which points at the cell interior, the nucleus, the RER, all of that sort of stuff, and the trans face, which points in the opposite direction towards the cell exterior, and just the plasma membrane, which is where it's going to export all its products to. So it receives them at the cis face, it exports them from the trans face. it can be a little bit difficult to tell which side is which. And so that's why I've included the micrograph here in the bottom right. You might assume that the side on top is the cis phase and side outside is the trans phase because of that curve, but it's actually the opposite way around. So it's very difficult to tell which one is which, and they won't always ask you. They'll usually include some sort of context so you can tell what's actually going on. Right, so we talked about vesicles, our little packages of whatever we need to transport within the cell. We've also got different subtypes of those. So lysosomes and peroxisomes are both organelles which contain digestive enzymes. Lysosomes, they just sort of have general digestive enzymes. So they break down whatever waste the cell needs to get rid of and biomacromolecules. So if you need to get rid of starch, you need to get rid of lipids, anything like that, you chuck it in a lysosome and it breaks them down. Peroxisomes are a little bit more specialized. So they oxidize a long chain fatty acids, think like palmitic acid and that sort of thing. And they also deactivate hydrogen peroxide, which is very important for you because hydrogen peroxide is toxic to you. So that's probably its most important purpose, and that's how it gets its name. We have a lot of them in phagocytes, which are cells which eat things like bacteria, because we basically use them to break down bacteria. In the little micrograph to the right, you can see a bunch of lysosomes and peroxisomes. You can actually see on a micrograph, there might be different shades of darkness depending on what stage they are in digestion. We also have secretory vesicles. So these are basically normal vesicles. And the distinction there is that they're purpose. So instead of transporting things within the cell, they transport things to the plasma membrane to be exported from the cell. Right, one thing that you don't cover very much in VC biology, but becomes quite important in med, is the cytoskeleton. So this is a big network of little tubes throughout your cell that basically go all the way through the cytoplasm. So it's important to think of it not like your bone skeleton, which the shape is pretty constant, especially once you reach adulthood, but this is constantly changing. So this can always move, it can always reform in different places. And even though it provides structure to the cell, it is a dynamic one, so it can change all the time. And there are 3 main different types of filament that we're going to talk about, and they all serve different functions. So we can move things throughout the cell using the cytoskeleton. It's almost like a scaffold. It obviously acts as a skeleton for the cell, and that's where we get the structural support. And we also have cell movement, which I'm going to talk about, which is a really interesting function of the cytoskeleton. And it can also be used for internal cell communication. So the little signaling molecules can move across it. All right, these are our 3 types of filaments. It looks really scary. Don't worry, it's not. So our first type and the biggest type is microtubules. So these are made of a protein called tubulin. And we have a little dimer. So basically just means we have 2 bits of tubulin. So we have the alpha tubulin type and the beta tubulin type. We squish them together, we get 1 dimer. And then we arrange the dimers in a big hollow tube. And there's 13 around, you don't need to know that, but it's quite a large diameter, 25 nanometers. That's not big for our scale, but it's very big in the cytoskeleton scale. The second type is microfilaments, and these are sometimes called actin filaments because that's the predominant protein involved in them. So we have 2 intertwined strands that sort of wrap around each other in a big helix shape. And these are smaller diameter because, like we said, they're not made to the same structure. These are 8 to 12 nanometers. We also have intermediate filaments. So unlike microtubules and microfilaments, which are usually made of the same sort of small group of proteins, these can be made of a lot of different proteins. You don't need to know the specific ones. You can just sort of treat them as one big group. And so they have a structure of a coiled cable, basically. Right, so microtubules, that's a big one, a hollow strand of tubulin. And the primary structure is to protect the cell against compression. So that's our skeleton, basically. If you try to smush a cell, this is what's going to help it keep its shape. But they also have more specialized structures. So you've probably heard of centrioles if you did mitosis at any point. And these are basically what makes up centrioles. We have 3 of them, and then we have 9 sets of those, which make a little pinwheel shape, if you look at the diagram over here. And those, their actual function is to make big spindles, mitotic spindles, and they pull chromosomes apart during mitosis. We'll talk a bit about that when we talk about cell proliferation, cell death, but that's the basic idea. We pull things apart in order to split the cell into 2 cells. Flagella are a very long structure, so they're like a big tail, And the cell, usually a bacterial one, can actually flick it like a whip to sort of propel the cell through some sort of watery medium. Cilia are very small, so they're like hairs around the edge of the cell, and they can do different things. So some bacterial cells especially can use them to move around like a bunch of little oars that they wave again and again. But we can also have non-moving cilia. So if you look there, there's a motile cilium on the left and a primary cilium on the right. And so those can serve as detectors for the cell. Now, the structure of all of these is the same basic idea, in which we have these little poking up things, and they sort of extend up into the cell membrane. So you can imagine if you tried to like poke your finger through cling wrap, it would sort of conform to your finger and be pushed up in the middle. That's basically what's happening here. So these are not exposed to the outside of the cell. They push up into the cell membrane and form these little appendages. And the structure of the latter two is where we have 9 pairs around the outside and then 2 solitary ones in the middle. So this is where a couple of those different structures look like under a microscope. We've got cilia over the left. Those are the little hairs all around the outside of this cell. This is a paramecium bacteria. And we've got the flagella. You can see how much longer it is. Like the cilia would be barely visible on this, but you can see this really long tail. And that's what it uses to move around. All right, microfilaments. So there are 2 different types. The actin type is found in pretty much every cell. The second type is myosin, which we pretty much only find in muscle cells. And so these serve a variety of different functions. So in muscle cells, obviously they're used for muscle contraction. And so they actually serve to pull the cell into a shorter shape to help contract a muscle. They're also involved in mitotic cleavage, which is basically when we want to split a cell into 2 cells during mitosis. These form a little ring around the middle of the cell and they pinch it into 2. And so that's how we get that sort of impetus to during telophase to actually split the cell apart. They're also important for movement of organelles and vesicles, because remember what I said before, this is a dynamic structure. It can move and change. And the last part of that is also related to movement and change, which is we can actually move the entire cell using certain microfilaments which are near the cell membrane. And this is how certain cells like immune cells actually get around. So you can see in this diagram here, this is a sort of a scan of a cell. The microfilaments are the orange ones. You can see them hanging out there. Green is the microtubules. So those are our bigger ones. So you can see they're a little bit more evenly distributed. And the intermediate filaments are not actually marked on here because they're a bit harder to detect. So this is how we actually move the cell with microfilaments. We start with our normal cell. We've got all our actin filaments or our microfilaments hanging around near the edge of the cell. The first thing we do is we depolymerize them. So basically we dissolve them and move them away from this side of the cell. What that does is it creates an empty space on one side of the cell. At the same time, we're going to repolymerize some. So these might be the same. These might be different ones which have been waiting around to be formed, but they're going to form on the opposite side of the cell, and they're going to push up the cell membrane like we talked about with the microtubules. They're going to push the whole cell membrane that way. And since we now have space on the opposite side, we can actually pull the whole cell with it. So the end result is that we have the same cell, same shape, but it's now moved to one side. And if you repeat that over and over again, you can move the entire cell. Now, on a cellular scale, this can be a pretty slow process, but to us, it happens really fast. You can find videos online of cells under a microscope doing this, and they move pretty quickly. All right, this is a big scary table. You don't need to worry too much about it because you don't need to memorize this at all. But intermediate filaments are sort of the waste basket. This is where we put all the stuff that doesn't fit into our microtubules or our microfilaments. So they help to maintain the shape of the cell and to anchor the cell contents. We have a whole bunch of different types, and some of them serve more specific functions. The important thing is, this is a quote from one of our lecturers last year. They are ultrastructurally similar, i.e. they all have that same coiled rope structure, but they're chemically distinct because they're all made of different proteins. All right, that brings us to the end of cells. The next topic is cell proliferation, cell death. If you have any specific questions, feel free to ask them now. Voice or chat, both is fine. We're good. Okay, we've got a bright bunch this year. Good stuff. Oh, sorry. Okay, we got a direct message. Okie dokie. 9+2 structure. All right, I will drag you back up there. Okie dokie. So we're looking at our diagram on the bottom right. This is relevant for flagella and for motile cilia. So basically any of those big outfoldings which can move. So when we say 9, we're talking about the outer ring, which is called a doublet microtubule. So that's a pair of microtubules which are sort of intersecting one another. And then our 2 is the group in the middle. So when we say 9+2, it's actually 20 in total, but they're arranged in 9 on the outside and 2 on the inside. Does actin make up microfilaments like cilia and flagella? Not quite, Ben. So microtubules, i.e. tubulin, that's the protein that we use for flagella and cilia. So actin do make up microfilaments, but that's not the filament used for flagella and cilia. Okie dokie. Can we explain the cell movement via microfilaments? Sure. Okay. So the basic idea again, I'll just sort of go over it one more time. We start with a bunch of microfilaments which are sort of keeping the cell in place. You can imagine them all pushing out on the outside of the cell, which is this little sort of a sphere. So when we want to move in a certain direction, the first thing we do is we dissolve some of the filaments on the opposite side. So where every part of the cell was being pushed out before, now that part isn't being pushed out, so it's free to be pulled in a little bit. At the same time, we push out on the opposite part of the cell, so really hard. So you can imagine like somebody inside the cell, like stopping to push on one side and starting pushing really hard on the opposite side. That's what happens when we repolymerize the filaments on the opposite side. And so if you're pushing one way and there's nothing preventing the cell from pulling in the other direction, then we have a net movement. So that's what we're showing in this little diagram. We're taking away the support on one side and we're pushing really hard on the other side to pull it forward. Yeah, somebody just said hamster ball. That is exactly correct. It's like a little hamster ball. We're taking away the support on one side and we're putting a really strong push on the other side. Hopefully that answers your question. Feel free to ask me to clarify if it doesn't. We also have a couple admin questions. So somebody asked me, I think right at the start, when do we get recordings for this session? So we'll try and put this up as soon as it's available. Like Zoom takes a little while to process recordings. So we'll see how long it takes to get that, but we'll put it up as soon as we get it. And the slides will go up as soon as we're done. Cell moves in the direction of microfilament density? Yeah, although remember this is a transitory phase. So basically the initial push is where we get that, and then by the time we stop moving, it's back to a very uniform density. So you're right, like by the time we get to uniform density around the cell, we're not moving. We move when we have more density on one side pushing out and less density on the other side no longer pushing out. Microtubules, doublets, not triplets, that is correct. Somebody asked me about the depth of the content. That's a really good question, actually. So like in med, it can be quite hard to know what depth you actually need to know for a given topic. And so we try to basically focus on the high yield content. So this is the stuff that's most relevant for exams, and we're trying to do a pretty comprehensive view at that. There might also be niche stuff. So we, in this limited time, we try and be really efficient, but we can't cover quite as much as lectures. So those are always going to have a little bit more information on specialized stuff that we can't quite cover. So like we'd recommend that you come to these to get like a really good idea of the most important stuff, and then also go lectures, go to like, stay in school kids, go to your lectures, go to your workshops, do all of those so that you get a really solid overall understanding. Any tips for note taking? I'll leave that just to the end because I just want to make sure everyone has the time to see the main content. And then if you stick around at the end, I'll stay on the call for a while and we can talk a bit about stuff like note taking. Alrighty, I'll pass it on to Marwan, who'll be presenting cell proliferation, cell death. Marwan, do you want me to keep this, or are you happy to present on your own computer?
I'm happy to present.
No worries. All right, I'll stop my share. Oh, that's the wrong button. Oh, sorry. Somebody else also asked, is this the content we'll be covering during the week? That is the intention. So we've got access to your timetable, and we're trying to basically set up each week so that you learn the stuff in this session that you'd need to know for the week. Sort of a head start for you guys.
So, hello everyone. Can you see the screen? Or great. So my name is Marwan. Today we're going to talk about cell proliferation and cell death. So as an overview, we'll first go through the two ways of cell proliferation, mitosis and meiosis. Then we will talk about the regulators and inhibitors of this process of cell cycle. Then we will talk about the non-disjunction events, and we will give some examples. Finally, we will talk about cell death. Just as a warning, English isn't my first language, so I can't even understand myself sometimes. So if you couldn't understand anything that I'm saying, feel free to stop me and ask to repeat. So firstly, why do cells proliferate? Why do we need proliferation in our cells? There are three main reasons. We need it for growth, replacement, or producing gametes. As the cell theory says that all cells came from pre-existing cells. So multicellular organisms are growing through producing more cells. Replacement, like every cells have a lifetime. After that, they die, or sometimes they can be damaged. So we replace these cells through proliferation. Finally, we produce gametes or sex cells in order to reproduce. There are two ways for proliferation, mitosis and meiosis. We'll first talk about mitosis, which involves the cell cycle. So the cell cycle is the process of growth and division to form new cells from a parent cell or pre-existing cell. This process can be repeated many times depending on the type of the cell. For example, in some types of bacteria, it can be repeated as little as every 20 minutes. In some yeast cell, it can be repeated from 90 to 120 minutes. For our cells, mammalian cells, it can take 20 up to 24 hours and more. So the cell cycle or mitosis, we can divide into three main stages: interphase, mitosis, and cytokinesis. Interphase has three stages: gap1 phase or G1 phase, synthesis phase or S phase, and gap2 phase. Mitosis also have four main phases: prophase, metaphase, anaphase, and telophase. So interphase, it's the longest period of the cell cycle at all, in general, and the G1 phase is the longest period of all of them. So generally in G1 phase, the cell just grows and doubles in size. So, the number of organelles will be doubled, as well as the size of the cell. Then the cell might go into gap0 phase, G0 phase or S phase. Cells that go into GAP0 phase are non-dividing cells, like nerve cells, and basically they just stay in G1 phase. They just grow and die. They will not divide. But most of our cells go into the synthesis phase or S phase, where the genetic material is to be doubled. So the number of chromosomes will be doubled because we will have two cells have the same amount of genetic materials. The last stage of the interphase, it's the GAP2 phase, where the cell continue to grow and it prepare the cell for mitosis, the next stage. And it also check for any DNA errors to be repaired or corrected. So mitosis and cytokinesis, the goal of mitosis is to produce two genetically identical daughter cells from one parent cell. And the purpose of mitosis is either to growth or to replace cell. So mitosis contains of 4 main stages: prophase, metaphase, anaphase, and telophase. We can remember them by PMAT, PMAT. Prophase can be divided into 2 stages: early prophase and late prophase. Actually, in some textbook, you can find late prophase named as pre-metaphase, but it's still the same. In early prophase, the replicated DNA begins to condense, and in the late prophase, nuclear envelope breaks down, chromosomes continue to condense. They will be fully condensed at this stage, and the spindle fibers begin to form. At the metaphase, at this stage, the chromosomes will be at their most condensed state. They will align down the equator of the cell, or the equatorial plate, or some call it I forgot it. Sorry. So it's the equatorial plate. It's just at the center of the cell. Also on this stage, the spindle fibers will attach to the centromeres of the chromosomes. And in this stage, metaphase, most of the chromosomal diagnostic can be done, the karyotyping, because chromosomes can be easily seen and stained. At anaphase, spindle fibers contract and separate the sister chromatids, and pull them to the opposite sides of the cell, the poles of the cell. Finally, telophase, the nuclear envelopes reassembles. Cell starts to pinch to form a cleavage furrow, and chromosomes start to condense into chromatins. So it's the combination between the DNA and histones. And the final stage, cytokinesis, which is basically the separation of the cell, the dividing of the cell. So we will have 2 daughter cells. The cleavage furrow continue to deepen. We can see here the cleavage furrow. And then finally, we'll have 2 separate daughter cells. Also, cytokinesis, it often starts before the ending of telophase. We now move to the meiosis. The difference between meiosis and mitosis in the goal is that meiosis will produce 4 haploid daughter cells. So 4 daughter cells that have the half the genetic material of the parent cell. The purpose is to produce gametes or sex cells. Meiosis has meiosis 1 and meiosis 2. Both of them have the same phase, but prophase, but the difference is in the number. So, before meiosis begins, DNA replication occurs to doubles the genetic materials, and we refer them as 2N. So in prophase 1, nuclear envelope breaks down, same as prophase in mitosis. Chromosomes start to condense, and the homologous chromosomes pair up with each other. So, for example, chromosome number 21 will pair up with chromosome number 21. So we will have 4 sister chromatids for each chromosome. And during this stage, crossing over occurs, which means swapping the genetic material between the homologous chromosomes. We'll talk about that later as well. Metaphase 1, chromosomes align down the equator of the cell, and the spindle fibers will attach to the centromeres of the chromosomes. Anaphase 1, homologous chromosomes will be separated because of the contraction of the spindle fibers, and they will move towards the opposite sides of the cell. Finally, telophase and cytokinesis. The nuclear envelope reassembles and the cells start to pinch, making cleavage furrow, and finally it will be divided into 2 haploid cells. So each cell will have half the genetic material of the parent cell, and we will refer them as N chromosome, contain N chromosome. Then we'll move to meiosis 2, but there will be no further DNA replication between meiosis 1 and meiosis 2. In prophase 2, the nuclear envelope will break down. In metaphase 2, chromosomes align down the equator of the cell, and spindles attach to the centromere of the chromosomes. During anaphase 2, sister chromatids will be separated, and it will move towards the opposite side of the cell, towards the poles of the cell. And finally, during telophase 2 and cytokinesis, the nuclear envelope reassembles, chromosomes starts to condense, and we will have 4 haploid daughter cells in total, because each cell gives rise to 2 cells. Then during the meiosis, because sperms or eggs need to differentiate from the produced daughter cells from meiosis, we refer the eggs as oogenesis and the sperms to spermatogenesis. They both produce via meiosis. They both have to differentiate because they need to change structurally, like the sperm need to have tail and head and everything. They both can't survive for a very long time if fertilization does not occur. However, the process of producing sperms or spermatogenesis is rapid and uninterrupted. However, the process of producing oogenesis is must be interrupted at one or more stages. Also, spermatogenesis has equivalent mitotic divisions, resulting in 4 equivalent sperms. However, it's not the same in fishes, where it produces 1 egg and 3 polar bodies. This chart probably will make it clearer, as the genetic materials and the organelles, also the cytoplasm, will be divided equally during meiosis of sperms. So we'll have 4 daughter cells have the same size, same genetic materials, same organelles. However, in the process of producing eggs or ova, the cytoplasm or the organelles will not be equally divided between the produced cells, which result in 1 ova and 3 polar bodies. The first polar body actually will go also under meiosis, will undergo meiosis and produce another 2 polar bodies. Then we'll talk about genetic variation. So we as human have different mechanisms to meet genetic variation. Some of these mechanisms happening during mitosis or meiosis. One of them is crossing over, which occurs during prophase 1, where the genetic material between the homologous chromosomes will be swapped. And then the independent assortment, where homologous chromosomes lined up and cross randomly during the metaphase during meiosis 1. And finally, the random fertilization, as each sperm has 8 million different combination of the chromosomes, as well as the ova or the egg. So 8 million multiplied by 8 million, we will have 64 trillion possible diploid combinations. So yeah, humans are very special. Then we'll talk about the non-disjunction events. Like it generally means that when homologous chromosomes fails to separate, or sister chromatids fail to separate during anaphase. So this will obviously result in incorrect number of chromosomes. Either we have more chromosomes or less chromosomes than we should have. And non-disjunction events, when it's occurred in meiosis 1, will result in worse outcomes, as we can see here, because all the daughter cells will have abnormal number of chromosomes. However, if it occurs just in meiosis 2, we will have 2 daughter cells that have the correct number of chromosomes. So now we'll have some examples of non-disjunction events that occurs in sex chromosomes. We have 2 examples, Turner syndrome and Klinefelter syndrome. Turner syndrome, where we have just 1 single X chromosome and no Y chromosome. Klinefelter syndrome will have 2 X chromosomes besides 1 Y chromosome. So we have an extra X chromosome. It's good to mention that people with Turner syndrome or Klinefelter syndrome, they can survive. Unlike most non-disjunction events, most of cases will not survive even more than 1 year. For autosomal non-disjunction events, we have Down syndrome, where we have 3 chromosomes, number 21, so trisomy 21. Edwards syndrome have 318 chromosomes, and Peytus syndrome have 313 chromosomes. The cell cycle have a number of checkpoints during the process, which are very important to prevent any errors that happen during the DNA replication or the cell cycle. These checkpoints are very important to avoid serious diseases that might happen because of these errors, such as cancer. During these checkpoints, it will first try to repair or correct the errors. If that didn't happen, then the cell will undergo apoptosis. It will commit suicide. We have 3 checkpoints during the cell cycle. The first checkpoint is between G1 and S phase, where they check for the appropriate side, the availability of nutrients and growth factor, and it will also check for DNA damage. The second checkpoint is between S phase and G2 phase, where it will check for any DNA replication errors. The third checkpoint is during the metaphase. It will check for the attachment of these spindles to the chromosomes, and it will also check for the completion of mitosis. We also have cell cycle regulators, as well as cell cycle inhibitors that we are going to talk about them next. There are 2 main classes of regulators. We have cyclins and CDK, which stand for cyclin-dependent kinases. They basically work where cyclin bind to CDK and activate them. Then CDK will activate associated proteins to continue the cell cycle. There are these regulators occurs as checkpoints in 3 stages. One of them between G1 and S phase, where cyclin D reacts with CDK4 to produce the complex of cyclin D CDK4 and produce cyclin E. And then cyclin E will react with CDK2 to promote the expression of cyclin E. Actually, it promotes the expression of cyclins that promote the expression of S phase. So this ensures that S phase cyclins and DNA synthesis enzymes will increase, and the molecules that inhibit the entry into S phase will decrease. Then the S phase control, the cyclin A produced from the G1 S phase control, will react with CDK2 to produce cyclin B. and phosphorylate targets involved in DNA replication. This will ensure this ensures that that each replication complex is activated and new complexes will not form. Finally, the regulators between GAP2 and metaphase control cyclin B reacts with CDK1 to form mitotic spindles and repair the cell for mitosis. Then we will have the inhibitors. We have two main inhibitors, P53 and retinoblastoma, RB inhibitor. They both proteins, and they are very important to stop or arrest the cell cycle. P53 is named after its size, 53 kilodalton. It arrests the cell cycle and prevent it from going from G1 to S phase. It's a transcription factor for P21 that inhibits cyclin E CDK2 complex. And approximately 50% of cancer are a result of malfunctions of P53. Retinoblastoma also, it inhibits the or arrests the cell cycle from going from G1 phase to S phase. But if it was inactivated via phosphorylation, genes can be transcribed to allow cell to enter S phase. Finally, we'll talk about the cell death. There are two pathways for cell death: apoptosis and necrosis. Apoptosis is the normal and programmed cell death. However, necrosis is unprogrammed cell death and unexpected. So apoptosis, as we said, it's a programmed cell death, so it happens due to natural reasons. It can be due extracellular signals or reasons like toxins, hormones, heat, radiation, viral infection. And it can happen due to intracellular signals, for example, DNA damage or change in intracellular calcium. We can see that there are 6 stages of apoptosis. First, cell will start to shrink, and cytoskeleton will be broken down. And then it will start to condense, and organelles start to come close to each other or to compact. The third stage, it's called pycnosis, where chromatin starts to condense. Then the nuclear envelope breaks down and DNA start to fragment. It's called karyopexis or fragmentation. And then the stage of blipping where cell membrane buds and its shape come like your hand. After that, the cell breaks into apoptotic bodies, as we can see here, which then finally will be phagocytosed or broken down. Here's compare between apoptosis and necrosis. We said that apoptosis is natural and programmed cell death, where necrosis is unexpected cell death. Necrosis is unregulated, unlike apoptosis, where it's highly regulated. In necrosis, cells burst. However, in apoptosis, cells shrink. Apoptosis do not cause any damage to the surrounding cells and cause no inflammation to a very minimal inflammation. However, in necrosis, it causes damage to the surrounding cells and it causes inflammation, very serious inflammation. Also, in necrosis, cell membrane integrity is lost, while in apoptosis, it will remain. Necrosis will produce dead tissue that requires often surgery to remove them. However, apoptosis, as we said, the apoptotic bodies will be broken down or phagocytosed. Sorry, and that's all I have. Sorry for that miss. Now I'll look into the.
No worries. Thank you so much, Mama. What we might do for that one, so usually we're asking questions right now. We might hold the questions until the end because we just have a couple more short things on cells and bodily fluids and a bit of the introductory HKS stuff. So we might try and get those out of the way now so that you guys can all stay and get the maximal content, and then we'll ask the questions at the end. Yep. Hopefully that sounds good to everyone. I'll hand it over to Deepak.
Because we're kind of running out of time, but alrighty. So cells and body fluids. Let me share screen. 1 second. Can you guys see that? Is it a yes? Yes. Okay, cool. Alrighty, okay, so cells and body fluids is the first physiology lecture you guys are gonna have, and physiology is basically, sort of like physics, a little bit of physics and chemistry. So it's stuff that is, you can think of it as the sort of like the fundamentals that leads to more stuff. So I highly recommend getting your head around it, trying to understand it rather than memorize it and all that good stuff. So we're gonna talk about diffusion, osmosis, dissociation, and everything in these objectives. Also, with what James said in the chat earlier, I highly recommend looking at what you think is high yield according to what you see in the objectives, because I think that's generally a good idea to get to what faculty wants you to know. So getting into it quickly, diffusion. So what is diffusion? It is the spontaneous movement of particles down the specific concentration gradient. And a concentration gradient is created when you have more of some particles on one side and less of some particles on one side, right? And when things get a little bit funky is when you got a semi-permeable membrane. So what we mean by that is that it's a membrane that only allows certain types of particles, solid or solvent, to move. And typically it's something that is non-permeable to the solute. That's usually what we refer to when we talk about semi-permeable membranes. And that's when we get something like osmosis. So what is osmosis? It is essentially the movement of water thanks to the solute gradient when there's a semi-permeable membrane. So if we look at this little diagram over here, we can see that in this beaker that you have more of the solute on the right-hand side and less of the solute on the left-hand side. So typically what would happen if the membrane in the middle was permeable is that the solids would just rearrange themselves through diffusion. So the solids would just move from right to left down the concentration gradient. However, if the membrane in the middle is non-permeable to the solute, then we're going to see the water rearrange itself rather than the solute. So the water is going to move from a region of low solid concentration to a region of high solid concentration to sort of dilute the water on the other side. That's osmosis. And here's a concept called osmotic pressure. So when we have this semi-permeable membrane and the movement of water, if we have this exact same scenario, but we push on the right-hand side to sort of prevent the water from moving, then we can calculate what is known as the osmotic pressure. The reason why this is sort of important is because if you push on the right-hand side and that prevents movement of water, then this applied force to stop osmosis is also equal to the osmotic, like sort of like the force that's pushing the water along. So osmotic pressure is the minimum pressure that stops osmosis, which is also equal to the osmotic pressure of the solution, meaning the pressure that's causing the water to move from the left-hand side to the right-hand side. I hope I'm not going too fast, but just chuck it in chat if I am. Alrighty, so we have a couple of different concepts regarding solubility and concentration in solvents. So molarity is something you guys probably have already heard about. So molarity refers to general concentration that we know it. So it's the number of moles of solute per unit volume of solution. And we're going to introduce another form of measuring concentration, and that is osmolarity. So what is osmolarity? Osmolarity is the same structure. So it's the number of osmoles of solute per unit volume of solution instead of the number of moles. And the difference between the number of moles and the number of osmoles is that osmoles are the number of moles of solute that contribute to the osmotic pressure of the solution. So the reason why this distinction is important is because of dissociation. So if I say that 1 mole of NaCl exists in a solution, right? We're gonna have to break it down to 2 osmoles of NaCl because sodium and chloride are going to dissolve in that solution almost fully. So 1 mole of NaCl equals 2 osmoles of NaCl. And since we have double the number of solute particles, we're gonna have more osmotic pressure, and that's why it's relevant. And in the example that I just gave regarding NaCl, usually it goes all the way from NaCl to Na plus Cl, but there are situations where the dissociation sort of doesn't go all the way, right? It sort of like stops kind of halfway. So you have this equation here, which is AB to A plus B minus. And if you should be aware of how to calculate the final osmolarity of a solution like this. So this is just taken from a screenshot of Chris Wright's lecture that his notes that he delivered. So essentially, I'm just going to go through the last part. So if only 75% of AB dissociates, we say that AB has a dissociation constant of 0.75. And the way we use 0.75 is by multiplying it to the concentration of AB, like AB times 2. So 1 mole of AB is going to give 2 moles of A and B, right? So 0.75 times 2 is 1.5 osmoles. So we're going to have 1 mole of AB providing 1.5 osmoles on the right-hand side. And since 75% of AB dissociates, we're going to have 25% of AB left over. So that's going to be 0.25 osmoles on the left-hand side, added to the 1.5 osmoles on the right-hand side that are created to give 1.75 osmoles in the final overall solution. Okay. So we talked about osmolarity as another form of concentration measure, specifically related to osmotic pressure. And there's another one called osmolality. You're not really required to calculate this, but you're just meant to know that it sort of exists and it's different to osmolarity. So that there's an L in the place of an R here on the right one, the one on the right. And what it essentially means is it's just another form of solid concentration, but it's in terms of moles per kilogram rather than moles per liter. So it's just solid concentration per unit mass of the solvent instead of the unit volume of the solvent. Typically, it's always going to be the same, but changes in density sort of changes it. But just keep in mind that this is different. Okay, so now we get to the stuff that is clinically relevant, and that is tonicity. So if you guys didn't get any of this, the one concept I want you to at least get away from this is the fact that osmolarity is a thing and tonicity is a thing. So tonicity is what we refer to as effective osmolarity. So what do I mean by that? So in this situation, right, with the little U-shaped test tube and the movement of water, so the reason why the water moved is because the solute on the right-hand side, it cannot move through the semi-permeable membrane to the left-hand side. Because if this semi-permeable membrane didn't exist, or it allowed for the solids to move, then the solids would just rearrange together. They would just rearrange in the water solution, and no water would move. And osmosis wouldn't occur, but diffusion would occur. However, since there's a semi-permeable membrane here, then we're going to see the movement of water. Therefore, the solutes present in the solution are called effective osmoles because they contribute to the movement of water and hence the creation of osmotic pressure. So tonicity is essentially the number of osmoles of non-diffusible solute per unit volume of solution. Non-diffusible means that it doesn't diffuse across the membrane, but it promotes the creation of osmotic pressure. And this is why it's super clinically relevant, because we get into situations where we see red blood cells or just body cells surrounded by fluids with different concentrations of different stuff, right? So let's say we put a red blood cell in a solution we call an isotonic solution. So an isotonic solution refers to a solution that has the same tonicity, which is effective osmolarity, on the outside and on the inside. So what that means is the number of solutes is the same. So you're not going to see any movement of water or any movement of solutes. So the cell remains in a normal state. If you put the red blood cell in a hypotonic solution, so hypo meaning low, tonic meaning the concentration of non-diffusibles, then you're going to see water entering the cell. Now, why is that? It's essentially because you're going to see water, since the concentration on the outside is less than the concentration on the inside, and since we're talking about tonicity, which is the non-diffusible solutes, meaning the water is going to move and not the solutes, then water is going to move from the side with a lower concentration of solute to an area of higher concentration of solute. And that's going to cause the cell to explode because you're going to have a bunch of water coming in that's going to mess up the structural integrity of the cell and it's going to lyse. On the other end of the spectrum, you see a hypotonic solution. So hyper meaning high, tonic refers to the number of non-diffusible solutes. So this means that this solution has a lot of non-diffusible solutes on the outside, right? So if it's non-diffusible on the outside, what's going to happen? We can't have these solutes rearrange and diffuse across because they're non-diffusible. So we need water to move to sort of fix the concentration gradient. So we're going to see water escaping the cell to go to the more concentrated outside to get that to dilute and be the same. So that's going to shrivel the cell, or it's another word is crenation. You might come across that. And that's essentially it. In a red blood cell, typically you see the calculation of 300 mosms or milliosmoles per liter. And from what we just talked about, a hypotonic solution would have a lesser concentration, isotonic would be the same, hypertonic would be more. And just this small little thing, which might be obstructed from your vision, it just says that you can have a isotonic solution, which is, wait, can I move this? Okay. So it's essentially saying that you can have an isotonic solution, which is not always isoosmolar. So what does that mean? So that brings together two concepts that we talked about. So that's osmolarity and tonicity. So tonicity refers to, okay, first of all, osmolarity refers to the number of essentially the number of solutes in the concentration. Tonicity refers to the number of non-diffusible solutes. So what this means is that the solutions can have a bunch of other stuff which increases its concentration, which makes it not isoosmolar. But if all of this bunch of other stuff is diffusible and can move across the cell membrane, it does not contribute to tonicity, and it does not contribute to the movement of water. So you can have stuff that is not isoosmolar, but isotonic. And that's an important understanding to have. Okay, so just a little bit of info on body fluids. So you typically kind of want to know what the general ions are that are predominant intracellularly and extracellularly. You're going to come back to this though, in terms of neurophysiology when you're talking about conduction of action potentials. But for now, just know that this outside is always generally salt water. So you see NaCl extracellular, intracellular, you see potassium and phosphate, and also hydrogen carbonate on the extracellular. This breakdown of body water is something that also you should know. So first of all, first thing is that 60% of body weight is generally the amount of total body water. And then it breaks down into basically 2/3, I mean, 2/3 and 1/3. So 1/3 is extracellular, 2/3 is intracellular. And from the 1/3 that's extracellular, 1/4 is plasma fluid, and 2/3 is interstitial fluid. So what I mean by interstitial fluid is fluid in between cells. Plasma fluid is fluid in the blood. And that's when we come to this part on the right. So blood is composed of plasma and cells. And there's another thing that you might come across, which is called serum. Serum refers to the pure plasma minus the clotting factor proteins. So you can essentially say that blood equals serum plus clotting factor proteins plus cells. Okay, moving on. So this is probably the most important table you're going to see from this lecture. And this is basically different scenarios. We're going to go through different scenarios and think about what's going to happen to the red blood cell. So this is us putting a red blood cell in pure water, putting a red blood cell in 0.9% saline, putting a red blood cell in dextrose and urea. So pure water, what's going to happen? So from the get-go, we know that it's hypoosmolar on the outside because pure water has no solid concentration in it. It's also hypotonic. Instantly, we know water is going to rush in because some ions inside the cell are non-diffusible. So water is going to rush in, and it's going to cause hemolysis, which is breakdown of the red blood cell. Next one, 0.9% saline. So these are also clinical situations. So it would be good to remember this. So we use 0.9% saline, and when we put in 0.9% saline, that roughly approximates 300 milliosmoles per liter. So that's an important thing to know. So it's 0.9%, not 9%, but 0.9%. Since it is salt water, NaCl, you might have seen this here. You should, on the cell, on the surface of the cell, you have sodium and potassium pumps, which ensure that the sodium and potassium concentrations are the same inside and outside. It sort of regulates it. And when sodium doesn't move into the cell because of that regulation, the chloride also sticks to the sodium and stays on the outside. And that's what I mean by it's effectively impermeable. Because technically there are channels that allow for stuff to come in, but because of this active process of pushing sodium out of the cell, the sodium is going to stick to the sodium because it's NaCl. And also usually stay outside of the cell for these purposes. So particles don't move, and we begin with equal osmolarity. The reason why particles don't move is exactly what I explained just then, because of the active transport. Equal osmolarity because 0.9 is 300 milliosmoles. And so water does not move because it's isotonic, because the tonicity in the inside, which is the non-diffusible, is the same as tonicity on the outside. So water injected in the solution remains exactly where it is. So when we go back to here, what we are going to think about is where does the water go, right? So if we inject it into the intravascular space, because we're going to put in blood, basically. So it's going to go into your plasma fluid, and then it can't enter cells. So it's going to essentially redistribute into the extracellular fluid. So you're just going to see an increase in extracellular fluid and intravascular space with no difference in intracellular fluid. So if this is like, say someone is bleeding a lot and lost a lot of blood volume, I think this is like, there are reasons why you would put extra water into some components of the body rather than others because of specific situations like that. Moving on to 5% dextrose / glucose. So 5%, again, it's 5% because it's also approximately 300 milliosmoles per liter, which is the same as the red blood cell and general cells. So, but this thing is a little bit special. So it is isoosmolar, similarly. And initially you have the same osmolarity inside and the outside. However, dextrose or glucose is slowly broken down in respiration. So what happens is that the extracellular fluid slowly becomes hypotonic until only pure water is left. The reason why it becomes hypotonic is because it slowly loses its tonicity because the glucose is being used. And then eventually it becomes pure water. So what you see is over time, since over time it turns into pure water, as hypotonicity increases, you're going to see movement of water into the entirety of the total body water. So it's going to start here, but then since it's slowly becoming hypotonic, you're going to see water move in to intracellular and contribute to total body water. It doesn't do, the reason why we use this rather than just injecting pure water is because when you inject pure water, that sudden influx is just going to enter cells like extremely quickly and burst them. So the reason why we use 5% dextrose is for it to slowly, you're essentially slowly creating pure water until more and more water flows into the cells, which eventually leads to increase in total body water. And there's no hemolysis as a result. It's a little clever trick. And finally, last but not least, 1.8% urea, again, isoosmolar, so approximately 300 milliosmoles per liter. But in this case, it's going to be permeable. So we're going to look through urea as this specific example, because I think this is another concept that's a little bit tough to grasp, but it's good if you do. So we've established that it has the same osmolarity. However, we know that it's permeable. So it's it's not a non-diffusible, it's diffusible, right? So it's not going to contribute to tonicity, but it's going to contribute to, it's going to be able to diffuse across the membrane. Therefore, it's not going to contribute to tonicity. Therefore, it's not going to contribute to movement of water. So that's why in the final column here, we have 300 for the cell and 0 for ECF. So technically, there's no water movement because water generally moves from area of low tonicity to high tonicity. But in this case, it's not going to do that. Wait. Wait, okay, hold up. No, hold up. Okay, I made a mistake there. So because it is 300 to 0, water is going to move. And water is going to move into this cells because it is hypotonic. Even though, so this is the example where it is not isotonic, but it's isoosmolar. Yeah, so this is going to have the same effect as injecting pure water, because once the diffusibles diffuse across and rearrange, what you're left with is essentially super hypotonic ECF, and then water just moves into the cell. So something that is important to understand is that the movement of osmolarity, the change in osmolarity, works like this. So since you have 300 milliosmoles in the cell, and there's a ton of, okay, so there's a ton of solute in the ECF bath, there's a very small amount of solute in the cell in order to get 300 milliosmoles per liter, because there's there's such little water in the cell. So when the solute moves from the when urea diffuses across the cell membrane to match the concentration inside the cell, there's so much urea outside the cell that there's negligible change in the ECF bath osmolarity. Okay, wait, yeah. So this is, the reason why it moves even though it's 300 and 300 is because this 300 is for something else, but this 300 is specifically for urea. Since the urea concentration inside the cell is nothing, because usually you don't have that much urea in the cell, urea wants to move into the cell, and it contributes to an increase in osmolarity of urea in the cell. However, tonicity does not change because it's a diffusible. You guys should look through Chris Wright's lecture notes, please, because he explains it very well. But feel free to ask questions later. But yeah, that's the end of cells and body fluids.
Yeah, thank you so much for that, Deepan. We got one more topic, which is the HKS one, which Raymond will be presenting for us.
Yeah, we'll do it. Awesome. Well, let me quickly pull up the share screen. Here we go. Oh, back to this one. Basically, HKS is health, knowledge, and society, and you're learning this because you want to see how health is applicable in a greater scheme to just learning the stuff and then treating patients, and how it's applicable in the greater scheme and how it intersexualizes groups. So to begin off, what is health? This is just the standard World Health Organization definition, which is a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. The strengths of this definition are that it covers every single aspect that could be out there, but it's also its weakness, because if it covers everything, it's also it doesn't define things that well either, so it's quite abstract, and that's where its flaws are at. So to talk about health, we need to first be able to measure it, and so these are some basic measurements. So first of all is life expectancy at birth. That's the number of years a person can be expected to live based on current death rates, but as we know, it's expected life expectancy, so it's not what you're always going to get. Then we have years of life lost, which is basically the theoretical number of years a person should live, but did not. However, this is not just, I died at 35, let me minus that from probably, I think right now it's 84.5, which is current life expectancy, and that is your years of life. It's not that, because it is a calculation that is weighted in relation to the whole population. If you look at this quick maths equation over here, bring back days of methods, basically it's the sum of the number of people in our population who die at specific ages, and that is added up and then divided by the total population, and then that gives you your calculated years of life lost. Moving on, we have years lost to disabilities, which is basically a calculation based on the condition that you get, this disability weighting and the length of the condition. This is, there's a table that is, you can find it on Wikipedia, or it's probably somewhere there as well on the internet, and basically it has given a certain value from 0 to 1, being 0 as healthy and 1 as dead, and it gives you 0.5, 0.6, 0.7, so on and so forth, and you throw it into the equation, which is incidence of condition, disability weighting, and times the average length of the condition to get your years lost to disability. And how these two ideas are relevant is the disability adjusted life years, which is the sum of years of potential life lost due to premature mortality, and the years of productive life lost due to disability, which is equal to the years of life lost plus the years of years lost to disability, as it's just basically what you would have lived plus the years that you had the disability for, and that gives you just basically the years that you could have lived healthily but didn't. Another idea to create that is basically, in addition to that, is incidence and prevalence. Incidence is how many new cases of X disease is appearing over a given time, such as COVID. You could be like, there's this many cases happening every day or something, and then the number of cases present in a community or world at a particular time, which is basically there is 5,000 cases of X at this point in time. Here's a nice diagram that is basically a good way to sum up incidence, prevalence, and mortality. Yeah, and over here is an example, and basically over here you can see the calculations of your disability adjusted life years. You can see here that for the years of life lost, they use a crude estimate here, where they just go by, they just take the number of people who died times the number of years they could have lived for straight away, instead of doing the huge equation that is required to actually get a more accurate answer. Yeah. Another topic that is going to be explored in further detail in your classes and through your lectures with Meiosis are basically equity and equality, and the difference between them you should know already, but to give you a refresher, equity is that everyone gets what they need to get to their full health potential, whilst equality, everyone gets the same amount. Like, this is the classic image to the right, which is basically people are watching a game. If everybody was equal, they would just get the same amount of support, and basically only some people will be able to see the game, whilst to the next on the image to the furthest to the right, you see that people are given what they need so they can all watch the game. Example of this is somebody living in rural Australia and they require chemotherapy. However, they are unable to access that in their rural area and they need transport to a larger city and that transport is provided. That would be creating equality, whilst somebody in a major city may already have access to it. Yeah, so it's basically leveling the field to get to healthcare. Another idea is how global forces interact and how they can affect people's ability to reach their complete health potential, which is the WHO's definition again. Things such as geopolitical forces such as conflict, displacement, and neoliberal globalization. These things all have big effects, and you'll be exploring them further within your studies. Social cultural factors such as human rights will, if they're not respected, it will lead to poorer health outcomes, as I think everybody understands. And there's also the determinants as well, as although it's great. Yeah, so we have health determinants and social determinants of health, so sex, age, race, and they all play a factor, such as if you're older, you're more likely to die, your sex, females are statistically said to live longer, and race also, but that's due to a lot of, that can be also traced back to environmental factors as well as geopolitical factors that happen in those regions that have resulted in their lower life expectancies and their poorer health outcomes. Environmental change can also result in big challenges for the health of those citizens, such as many of those island nations that are being inundated by waters as rising sea levels continues to occur. And also there's historical influences such as colonization. As you can see in basically all parts of the world, the First Nations people are always marginalized and they face discrimination. And because of that, they have poorer health outcomes, as I think everybody would understand, because because of discrimination and problems within the health sector that prevents them or has caused them to get poorer access to healthcare, and as a result, poorer health outcomes. And I think that is it for me.
Yeah, that brings us to the end of today. So thank you so much for coming, and thank you so much for sticking it out. I didn't have this amount of patience in year one. You guys are absolute champions for sticking around and listening to it. we've done our best to give you a good overview of basically what's coming. So yeah, this is like 1 week's worth of the content. There's a lot of content in Med. So we're going to try and condense it down as much as we can. So like obviously in lecture form and workshop form, this is like 10, 20 hours or more of content. So the one thing I'll try and get across to you guys before you guys head off is that we are going to try and do our lessons at 10 a.m. on Mondays because we don't have to take up too much of your day and like run too late. So we've shifted today because you guys had that Zoom, but assuming that you guys don't have anything else on Monday mornings that would preclude it, we'll be going at 10 a.m. Mondays from next week onwards. Thank you all so much. Feel free to stick around. We're going to be answering questions for a little while if you have anything you'd like to clarify. But otherwise, thank you so much. See you around. Oh yeah, one more thing, sorry. We're going to try and get you guys some like revision materials. We'll try and make some stuff for you guys to help you like test your knowledge and all that. So keep an eye on the Facebook group. That will be like our main mode of communication. Have a look there. Love you all. See you around.
Thank you so much.
Thanks for coming, guys.
Thank you.